Hello and welcome to BioCyc. This webinar will introduce you to the information present in BioCyc and to the many tools available to help you search and analyze information in BioCyc. Our overall mission at BioCyc is to provide you with up-to-date and comprehensive information on microbial genomes, metabolic pathways, and regulation, together with powerful bioinformatics tools for searching, visualizing, and analyzing that information. BioCyc is a web portal and a database collection that integrates genome and pathway information with powerful bioinformatics tools. The Cyc in BioCyc stands for encyclopedia because it's an encyclopedic reference with 18,000 total databases for different sequenced genomes. Of those 18,000, 60 of the databases are curated, with their contents derived from 130,000 publications. And you can refer to this URL here to see which are the curated databases. It integrates a really broad array of data, and its use cases include information finding, transcriptomics data analysis and metabolomics data analysis, sequence analysis, comparative analysis, and metabolic engineering. The creation of BioCyc databases follows this general model right here, where we obtain sequenced and annotated genomes from the RefSeq component of GenBank. We then apply a series of computational inferences, data import operations, followed in some cases by curation of the database. Computational inferences include prediction of metabolic reactions, transport reactions, and metabolic pathways, and prediction of operons. Data import includes importing of regulatory data from RegTransBase and of Go terms and protein features from UniProt. For those databases that we do curate, curation covers a number of different tasks, including correcting annotation errors when known from literature-based functions of genes, performing literature searches for new gene functions and pathways, defining protein complexes, authoring mini reviews for genes and pathways, updating a variety of database fields, and entering new enzyme reaction associations and new metabolic pathways. We also, for some databases, define a number of regulatory sites, such as promoters and transcription factor binding sites, as well as defining regulatory processes. Our E. coli database is the most highly curated database in BioCyc, with its contents derived from more than 40,000 publications. It contains many dimensions of information about the E. coli K12 genome, including the mini reviews that we author for genes and pathways and associated literature citations, gene ontology annotations, protein features such as active sites, metabolic pathways and their component reactions and metabolites, as well as regulatory information and gene essentiality data sets. The tools present in BioCyc span a number of different areas. One is Genome Informatics, where we provide extensive gene and protein search operations as well as genome site search operations. There's a gene information page with extensive information about genes and their gene products, and with the ability to retrieve the sequence of a gene or a genomic region. There's a transcription unit page that shows regulatory sites and interactions for a given operon. There is a blast search, sequence pattern search, and sequence alignment operations. There is also a genome browser that works both in a linear mode and a circular mode. It can zoom from the full chromosome in one window to the sequence level. It has a comparative mode shown here, where different chromosomal regions can be aligned at the same gene. And it has omics data painting capabilities. The simplest way to search BioCyc is to first select your current database to search of the 18,000 available, and then enter a gene name, protein name, metabolite name, or pathway in this quick search box here. So let's say I've entered the gene name SINR in B. subtilis. I'll get the SINR gene page, which in its initially displayed tab shows me all the proteins that regulate the transcription of this gene. This is a more complex query tool where the user can search for genes and gene products according to many different criteria, including the gene name or database identifier, protein name or identifier. We can also filter by sequence length and by genome map position. In this case, we've asked for all genes whose product is less than 50 amino acids long in the first million bases of the chromosome. And there are a variety of other searches available, including searching by PI, by the number of subunits of the complex, and by gene ontology terms and existence of protein features. There are a variety of pathway informatics tools available, including pathway search and a pathway information page, reaction search and a reaction information page that shows reaction atom mappings. There's a metabolite search and metabolite information page, and the metabolite searches span multiple different properties of metabolites. There's a zoomable organism-specific metabolic map diagram and a metabolic route search tool that computes optimal paths within the metabolic network from a starting compound to an ending compound. And we also have quantitative metabolic flux models available for a handful of the genomes in BioCyc. Here's an example of a pathway page in Synechococcus elongatus. And the reactions and the metabolites and the enzymes are all clickable. If we click on a metabolite, we'll go to a metabolite page such as this one, and clicking on the reactions tab will show us all of the reactions that consume the compound or produce the compound organized by pathway.